The overall goal of this procedure is to identify small molecules that are capable of disrupting the interaction between TRPA8B and HCN channels.
This method identifies candidate molecules that could be developed into new treatments for depression. The main advantage of this technique is that it can be completed in a high-throughput fashion.
This technique has implications for the development of novel therapy for major depressive disorder, because our lab has shown that disrupting TRPA8B binding to HCN may be of therapeutic benefit.
Though this method was developed with depression in mind, disrupting the interaction between TRPA8B and HCN may be of therapeutic benefit in other disorders such as chronic pain.
Visual demonstration of this protocol is important because it illustrates the benefits of using automated, high-throughput instruments to advance basic and translational research at open facilities like Northwestern University's High-Throughput Analysis Lab.
After purifying the TRPA8B241602 protein fragment and examining protein-protein interactions with a positive control according to the text protocol, thaw aliquots of TRPA8B and HCN1 FitC and keep them on ice. Use FP buffer to prepare 20 milliliters of 2 micromolar TRPA8B and 50 nanomolar HCN1 FitC. Then dispense 25 microliters of the mixture into each well of a low-binding 384-well black microtiter plate. For library screening, use an acoustic liquid handler to dispense 100 nanoliters of compounds into each well of columns 3 to 22.